please put them in the comment section below. And also remember to subscribe to my YouTube channel, Carrie Donegan. I'd appreciate that. And we'd love to talk to you as much as possible. And YouTube is a great way to do that. So now that we are live on both platforms, let's get started. The premise of this chapter really talks about why the immune system does what it does. Do we really need to take our immune system and boost it? Do we need to run to the far corners of the world looking for a secret herb or fruit or juice or potion or traditional remedy to make sure that our bodies don't fall into illness and disease? Or is it normal? Is it natural for the human body to get sick from time to time? And if it is natural, then why do some people succumb to certain diseases and get very sick and even die? And other people seem to weather the storm pretty well. Maybe they once a year end up with a cold or a flu that they can actually muster through and even get to work with. Why is it that some families have children who are full of allergies and other families seem to have no problems with allergies whatsoever? Well, we know that there are two types of cells that we're focused on, and not to get too sciencey on you, but just so that you know, there are the T helper cell ones. They're the things that deal with bacteria and viruses. And then there are T helper cell twos. And those are the things that we find help with parasites and worms. Now, interestingly, people who tend to have allergies have an abundance of T helper 1 cells, and people who have autoimmune diseases have an abundance of T helper 2 cells. So is there a connection or a relationship there? Well, we don't have the answer concretely yet, but we do want to make sure that we have to understand the background of where all of this is coming from and where we're going to be kind of diving into. And it brings us to this interesting concept, and that is that you are a holobot. And what is a holobot? A holobot is an environment where the animal or human, right, is in relationship with its microbiome. And I want you to think of yourself not so much as 100% human, but as Alana Collin is proposing, 10% human and 90% microbiota, which, if you really think about it percentage-wise, makes us a vessel that contains a vast microbiome. Not the other way around. It's not that the human body is the prevalent part of who you are. You are actually mostly made up of your microbiota. So let's dive into this chapter a little bit. I want to actually read it to you because some of the science can be a little bit dense. And we're going to move through, now that I've set up some of her premise of where we're going, and see where this information takes us. So she says, it seems paradoxical. Surely antibiotics are there to treat infection, not cause them. But although a course of antibiotics might cure one infection, they may also leave us open to others. The obvious explanation is that without protective microbes, the body is left exposed to attack by pathogenic ones. But antibiotics rarely decrease the total number of protective microbes present. Instead, they shift the composition of the different species. It seems the real change is in how the immune system behaves, according to which members of the microbiota are present. The first line of defense in the gut is a thick layer of mucus. Close to the cells of the gut lining, the mucus is free of any microbes. But in the outer layer, many members of the microbiota make their home. Treatment with, for example, the antibiotic metronidazole kills just the anaerobic bacteria, those that live without oxygen. This shift in composition of the microbiota changes the fundamental behavior of the immune system. It interferes directly with our human genes, dialing down production from the genes that churn out mucin proteins for the protective mucus layer. With a thinner layer left behind, microbes of all kinds have easier access to the gut lining. If they or other chemical components get through the bloodstream on the other side, the immune system is mobilized. You'd be forgiven for wondering if antibiotics change the functioning of the immune system, wouldn't we be getting sicker as a result of taking them? In a study of 85,000 patients, those taking long-term antibiotics for the treatment of acne were more than twice as likely to suffer colds and other respiratory tract infections as acne patients who were not on antibiotics. Another study of college students found that being on antibiotics quadrupled the risk of getting a cold. But what about the effect of antibiotics on allergies? In 2013, a group of scientists at the University of Bristol in the UK asked just that question. They used a massive research project known as the Children of the 90s. This project collected a huge amount of health and social information about the children born to 14,000 women who were pregnant in the early 90s. The data included information about antibiotic use when the children were infants. It turned out that those who had been given antibiotics before the age of 2, a startling 74% of them were on average nearly twice as likely to have developed asthma by the time they were 8. The more courses of antibiotics the children received, the more likely they were to develop asthma, eczema, and hay fever. But as the saying goes, correlation does not always mean causation. The lead researcher on the antibiotic study had discovered 4 years earlier that the more television children watched, the more likely they were to develop asthma. Of course, despite similar results in the antibiotic study, no one really believed that the act of watching television could bring about the immune dysfunction in the lungs. In fact, the number of hours in front of a television was being used as a proxy for the amount of exercise children were getting. The point still stands, though. How can we be sure that the amount of antibiotics a child received isn't also a proxy for something? For example, it may relate to how fussy the parents were, or perhaps more pertinently, the possibility that antibiotics were given to treat early symptoms of asthma. But the researchers accounted for this by calculating the likelihood again, this time excluding any children who had suffered from wheeze before the age of 18 months. The link remained strong. Of course, taking antibiotics is all about ridding the body of infection, so the hygiene hypothesis stand up in light of the link between them and allergies. But the paradox remains. Why would the immune system attack harmless allergens in preference to apparently more alarming threats from the body's microbes? And if the rise in allergies were connected to the drop in infections, why wouldn't those of us who had fewer infections be the ones that had more allergies? Professor Agnes Wold of the University of Gothenburg in Sweden was the first to provide an alternative to the hygiene hypothesis in 1998. Research into the importance of the microbiota was just beginning to pick up steam, and the lack of correlation between infections and allergies had started to challenge Strickland's idea. Alongside the correlation between antibiotic use and allergy was a more direct connection. Wold's colleague Ingrid Elderberth had previously compared the microbes of babies born in hospitals in both Sweden and Pakistan. The babies born in Sweden, where allergy rates are high, had a much lower diverse bacteria than the babies born in Pakistan, especially a group called the Enterobacteria. For sure, Sweden's level of hygiene far exceeded that in Pakistan. But these babies weren't getting sick. They weren't suffering from infections. They were simply being colonized by more microbes, especially from a group of bacteria that are found in the adult gut. And therefore, the mother's feces, as well as the general, environment. Midwifery protocols in Sweden included, in some cases, practicing of cleansing of women's genitals before they gave birth. Many have been utterly altering the microbes. They set up camp in the empty guts of newborns. Wold felt that it was this change in the composition of the microbiota rather than the exposure to the outright infection that was driving the rise in allergies. She organized a large study of babies in Sweden, Britain, and Italy, and tracked the changes in their microbiomes over time. As expected, the ultra-hygienic babies were colonized by fewer species, again, especially of the enterobacteria. In their place were more members of a group of bacteria that typically are found on the skin, not in the gut, the Staphylococci. No single species or group of microbes could be linked to the development of allergies later in life, but the overall diversity of microbes in the baby's guts could be. The babies who went on to develop allergies had far lower diversity on their guts than the babies who remained healthy. Wald's reframing of Strachan's original hygiene hypothesis had gained ground amongst immunologists and microbiologists. 2 decades of research into the body's microbiota have another layer of complexity to our understanding what triggers the immune system to develop a healthy way. It is not simply infectious disease that a super clean environment put a stop to, but normal colonization by the microbes that are sometimes referred to as old friends. These old friends have evolved with every step of the way, deeply engaged in the conversation with the immune system as they have gone along. The hygiene hypothesis has mutated into the old friends hypothesis, a new take on an old idea. Exactly what the microbiota is telling the human body, or the body of any animal for that matter, is the next question. How does the body know which microbes to trust and which are true imposters? The immune system operates a team of different cell types, each with a specific role in detecting and destroying threats, much like the armed forces, whereas macrophages are foot soldiers, which gobble up threatening bacteria, and memory B cells are snipers trained to attack a specific target. T helper cells, such as Th1 and Th2, are communications officers alerting other troops to an invasion. Triggering all of these responses begins with antigens, small molecules on the surface of a pathogen that identify it as an enemy. These antigens act like little red flags that the immune system automatically recognizes as danger signals, whether it has encountered that particular pathogen before or not. Since all pathogens carry these flags, they will always be detected once they enter the body. Back when the concept of self and non-self ruled the thinking of immunologists, it was thought that pathogens gave way their presence through the antigens that they wore on their cell surfaces. But what researchers didn't acknowledge at the time was that beneficial microbes also carried these flags, and that they sent them the same messages to the immune system as those pathogens. The flag merely identified them as microbes and told the body nothing about whether they were friend or foe. The microbiota was not simply being ignored by the enlightened immune system. Instead, our beneficial microbes must have found a way to persuade the immune system into letting them stay. You might think that all immune cells would be out to destroy targets and hunt for threats. But as with every system in the body, there must be a balance, dials that turn up and dials that turn down. It is no different in the immune system. Pro-inflammatory or attack messages must be balanced by anti-inflammatory or stand down messages. The anti-inflammatory side of the equation is brought about by a newly recognized immune cell, the regulatory T cell, which acts as a brigadier coordinating the overall immune response. Known as Tregs, pronounced T-regs, these cells exert a calming influence over the aggressive, bloodthirsty members of the immune system's rank. The more Tregs, the less reactive the immune system, and the fewer, the more aggressive it responds. Indeed, in children who have a genetic mutation that prevents them from producing Tregs, a catastrophic disease known as IPEX, immune dysregulation, polydactrinopathy, entropathy, X-linked syndrome results. The immune system is pushed off balance and it produces massive quantities of pro-inflammatory immune cells, causing lymph nodes and spleen to swell with excess. The aggressive cells fatally attack the body's organs, usually bringing on early diabetes, eczema, food allergies, inflammatory bowel disease, and intractable diarrhea during childhood. These multiple immune and allergenic disorders result in early death and successive organs are destroyed. The surprising new evidence, though, is that the commander-in-chief commanding the anti-inflammatory Tregs is not a superior human cell type acting solely on the body's best interest. Rather, it is the microbiota that delivers the orders using the Tregs as its pawns. But manipulating the numbers of the suppressive ranks of cells, they ensure that their own survival. For them, a calm, tolerant immune system means an easy life without fear of attack or elimination. The idea that our microbiota has evolved to suppress our immune system for its own benefit seems a little intimidating. A safety mechanism that fundamental to our survival in the face of attack by our oldest adversaries is being tampered with the highest level. But we needn't worry. Our long co-evolutionary history with our microbiota has fine-tuned the immune balance to the best of the advantage of both us and it. More concerning is the loss of diversity that has hit the microbiota of humans living in a Western lifestyle. With lower diversity, what becomes of the Tregs? A group of scientists, including Agnes Wold, asked this question using the laboratory's old favorite, germ-free mice. They measured the effectiveness of Tregs from germ-free mice and compared them with normal mice. In the germ-free mice, a far greater ratio of Tregs to attacker cells was needed to suppress the aggressive immune response, showing that Tregs produced in the absence of a microbiota were much less powerful than those in a normal mouse. In another experiment, simply adding a normal gut microbiota to a germ-free mouse increased the number of Tregs the mouse produced, and this calmed the immune system's aggressive behavior. So how do the members of the microbiota do it? Their cell surfaces are covered in red flags, just as found on pathogen microbes, yet they manage to pacify rather than irritate the immune system. To do so, it seems that each beneficial species may have its own password known only to it in the immune system. Professor Sarkis Mazaman, the California Institute of Technology, discovered such a password produced by a bacterium called Bacteroides fragilis. These species is one of the most numerous members of the microbiota, which often takes up residence in the gut immediately after birth. It produces a chemical called polysaccharide A, or PSA, which is released from the surface of B. fragilis in tiny capsules. The capsules are engulfed by the immune cells in the large intestine, and the PSA inside triggers the activation of Tregs. The Tregs then send out calming chemical messages to the other immune cells, preventing them from attacking B. fragilis. By using PSA as a password, B. fragilis converts the pro-inflammatory response of the immune system to an anti-inflammatory response. It is likely that PSA and other passwords produced by the early colonizers are important in calming the immune response and preventing allergies. Many varieties of these passwords have probably evolved to be produced by individual strains of microbes to gain acceptance as a member of the exclusive club within the human body. Just as in the fatal immune disease IPEX, animals with allergies are deficient in Tregs. It seems that without their calming influence, the handbrake is off and the immune system goes full speed ahead, attacking even the most innocuous substances. Let me now tell you about cholera, the disease behind liters of water white diarrhea that contaminated the water supply of Soho back in 1854, which continues to cause outbreaks of misery in developing countries today. It's caused by a nasty little bacterium called Vibrio cholerae, which colonizes in the small intestine, but it never intends to stay for long. Where most infectious bacteria attempt to sneak past the immune system until they have fortified their ranks enough to resist attack and cause persistent infection, V. cholerae flaunts its presence from the moment it arrives. The first phase of the mission, V. cholerae, attaches itself to the intestinal wall and reproduces as fast as it can. But rather than hanging around and causing permanent infection, this bacterium has other ideas. It gets out. It does so in vast numbers, mixed with a slurry of watery diarrhea it drains from the body of its host. Diarrhea is a strategy of both the bacterium and the immune system. The bacterium uses it as a means to get out and infect further hosts. and the immune system uses it to flush clean of the pathogen and its toxins. The mechanism is this: the intestinal wall is rather like a brick wall, in that a layer of cells packed closely together. But instead of using mortar that cements the cells permanently to one another, they are bound by chain like proteins. This makes the wall a little bit more flexible. Most of the time, anything aimed to get from the gut to the bloodstream is forced to try to pass through a cell, where it is subjected to all kinds of interrogation. But occasionally, the protein chains can loosen, allowing substances from the blood to the gut or vice versa. If necessary, water can flood from the blood between the cells and the gut lining and into the gut, causing diarrhea. Useful if the body ever needs to flush out a pathogen. Two things are remarkable about V. cholerae watery exit strategy. One of which is permanent to the impact of our particular microbial colonies and the workings of our immune systems. The other, with which I will start, is just plain interesting. It concerns the conversation that V. cholerae bacteria have with one another before they make their departure. I'm not being anthropomorphic here. These bacteria and many others besides really do converse. The infective strategy that many others besides really do like this. The phase 1, infect the small intestine and reproduce like rabbits. Phase 2, when the population has reached a certain size and the host is on the brink of death, get out in a torrent of diarrhea, ready to infect tens of new hosts. The difficulty with the strategy is how to know when the population is large enough to justify jumping ship. The solution is called quorum sensing. Each bacteria is constantly releasing a tiny quantity of a chemical. In the case of V. cholerae, it's called cholera autoinducer 1 or CAI1. The more bacteria in the population, the greater the concentration of CAI1 that surrounds them. Hence, the same stage of quorum is reached. That is, the threshold minimum number of members are present for voting. With this, the bacteria senses it's time to leave. The way they make this work is just as slick. The concentration of CAI1, along with another autoinducer AI2, cause V. cholerae to change their gene expression. Collectively, they switch off the genes that help them to stick into the intestinal wall. They then switch on the set of genes that produces substances that forces the intestinal wall to open the floodgates. One of these genes codes for a toxin called Zunella occludens toxin, ZOT for short. It was discovered by the Italian-born scientist and gastroenterologist Alessio Fasano, who is based at the Massachusetts General Hospital for Children in Boston. ZOT works by loosening the chains holding the intestinal cells together, allowing water to pour in and the V. cholerae to ride their way through the intestines to freedom. Fasano's discovery got him thinking. If ZOT was a viral key that fitted a human lock, what was the lock even doing there? Could there be, I have to get my computer cord or we will lose each other. Talk about mutual relationship. There we go. Electricity is important. Fasano's discovery got him thinking. If ZOT was a viral key that fitted a human lock, what was the lock even doing there? Could there be a human key that was meant for that lock that had been copied by the V. cholerae? Sure enough, Fasano discovered a new human protein just like ZOT, which he named zonulin. In the same way as ZOT, zonulin interferes with the chains binding the intestinal cells to one another and so controls the permeability of the intestinal wall. More zonulin means the chains loosen and the cells move apart, allowing larger molecules to pass in and out of the blood. Fasano's discovery of zonulin brings us to the bit that's pertinent to immunity. In the presence of a normal, healthy microbiota, the chains are locked in place and the cells are tightly bound. Nothing large or dangerous can get through the blood. But when the microbiota gets out of balance, it acts like a bit like a mild version of V. cholerae, irritating the immune system. In response, the immune system tries to defend itself, by releasing zanulin to loosen the chains, uncouple the cells of the intestinal wall, and flush out the system. The gut lining is no longer an impenetrable wall keeping out everything but tiny food molecules. Instead, it has grown leaky. Through the gaps between the cells seep all sorts of illegal immigrants making their way to the promised land of the body. Now, this takes us into controversial territory. The concept of leaky gut is a favorite of the alternative health industry, which can be as rapacious and truth distorting as its more mainstream sibling. Big Pharma claims that leaky gut syndrome is the root of all illness and many other evils besides, as are old as the industry itself. But it has not been until relatively recently that any scientific understanding of this cause, mechanisms, or consequences have been scrutinized. Though Big Pharma has many faults of its own, alternative medicine can best be summarized as using two kinds of treatments: those that don't work well enough to merit the title medicine, and those that are not yet backed by science and clinical evidence. Perhaps a third category of treatment deserves mention: those that cannot be patented and sold, including rest and a good diet. The scientific and medical establishment are wary of the concept of leaky gut. For patients fed up with not getting answers about persistent fatigue, pain, tummy trouble, headaches, and so on, leaky gut offers a believable explanation. It's something that both well-informed and well-meaning alternative health practitioners and charlatans jumping on the bandwagon can easily offer their patients as a diagnosis, with a reasonable set of lifestyle recommendations as a cure. There are even tests that patients can take. When properly administered, measure the degree of leakiness. So alternative health practitioners are able not only to give a measure of the problem, but to track improvements. But for medical doctors, there's little evidence filtering down through health departments and medical schools in support of the concept, and its credibility remains low. The UK's National Health Service website offers little encouragement to patients exploring the idea. Exponents of the leaky gut syndrome, largely nutritionists and practitioners of complementary and alternative medicine, believe the bowel lining can become irritated and leaky as a result of a much wider range of factors, including an overgrowth of yeast or bacteria in the bowel, a poor diet, and the overuse of antibiotics. They believe that undigested food particles, bacterial toxins, and germs can pass through the leaky gut wall and into the bloodstream, triggering the immune system and causing persistent inflammation throughout the body. This, they say, is linked to a much wider range of health problems and diseases. The above theory is vague and still largely unproven. But this view is quickly becoming outdated. An unwillingness to align with the alternative health perspective and risk hard-won credibility is perhaps putting off scientists and doctors who are involved in research regarding intestinal permeability and chronic inflammation. Even as a science writer, I'm hesitant about this broad topic in a book based rigorously on scientific research for fear of pushing more skeptic readers out of their comfort zone. But the research base is growing and the mechanisms are being uncovered. Remember, this is an old book, so a lot of this research actually has been done that she's talking about, hoping will be done in the future. Before you make your decision about whether this is quackery or reality, let me review the evidence. It starts with Alessio Fano and Zanulin. While his intention was to learn more about cholera's invasive tactics, he ended up realizing he had answers to another problem much closer to home. In the 1990s, Fasano was a pediatric gastroenterologist newly arrived in the States from Italy. Among his patients were children who had celiac disease and had relatively uncommon then, not even mentioned in a major 800-page report on digestive disease published in 1994. Wow. The children Fasano was treating would become very sick if they ate even a tiny amount of gluten. Gluten is a protein found in wheat, rye, and barley grains that makes bread dough stretchy and holds on to the air bubbles produced by yeast. In celiac disease, this protein brings on an autoimmune reaction. Immune cells treat the protein as if it were an invader, producing antibodies against it. These antibodies also attack the cells in the intestines, causing damage, pain, and diarrhea. Celiac disease is quite a special autoimmune disease. It's the only one for which the trigger is known, gluten. This knowledge kept the immunologists happy. They knew what was causing it. Geneticists were also happy. Lots of genes had been located to make some people more susceptible to celiac disease than others. Bad genes plus an environmental trigger equals disease, they thought. But as the gastroenterologist, Fasano wasn't happy. For gluten to cause disease, it had to make contact with immune cells. But to do so, it must cross the gut lining. For exactly the same reason as diabetics must inject insulin rather than swallowing it, gluten cannot do this. It's simply too big a protein to cross the gut while by itself. But Fasano's discovery of cholera toxin Zot and the human equivalent zanulin proved to be the clue he needed. In people who develop celiac disease, whether they are 8 or 80 years old, gluten is a bad set of genes are not enough. Something had to let the gluten through. Fasano knew that the intestinal walls were leaking in the celiacs, and he had a hunch that the zanulin might have something to do with it. He tested the intestinal tissue of children with celiac disease and the children without. As he suspected, the celiacs had much higher levels of zanulin. Their intestinal walls were opening up and letting gluten proteins through the blood. Were they triggered in an on an immune reaction? Now, about 1% of the population in the West suffers from celiac disease. All right, we've run out of time, so I have to put a pin in it right here on page 140. I would encourage you finish the rest of the chapter. There's only 6 pages left, and it's well worth your discovery. But I think it's fascinating the journey that she's taking us on. I also think it's fascinating that she's really opening up some questions that I think for a lot of us, we really thought, oh, we've already figured that out. Or, that's not something that I'm worried about because I don't have the genetics. Whatever it is, I think it's important for us to remember that we can all become susceptible to this if we don't feed our good microbiome with really, really good foods, fermented foods, and probiotics. This is super important. And for those of you who are not familiar with doTERRA, I would highly recommend a brand new product called Restore, which is full of an array of great microbiota. It's been a true pleasure to be with you. I am on this journey with you. We are going to find out and discover together and come up with solutions for health and longevity. I'll see you next week.